Hi, my name is Javier Jarazo, and in this video I'm going to present some of the work carried out in the context of the Horizon 2020 project called SysMedPD. I'll focus on a small part of the project, the modeling of Parkinson's disease in the lab. This part played a central role in the project, where a succession of work packages leads to the identification of candidates for the treatment of Parkinson's disease. This part of the project was carried out between the LCSB, Mimetas, Mitochondrial, and the University of Leiden. Before digging in the project, I'll give a short explanation about Parkinson's disease. Probably most of us are familiar with the motor symptoms associated with the disease, such as tremor and postural instability, but there are also non-motor symptoms, such as anxiety, depression, dementia, and anhedonia. And all of this is caused by the loss of a specific type of neuron, the dopaminergic neuron, in a part of the brain known as substantia nigra. There are known risk factors that increases the chances of developing the disease, such as age, environmental factors, and genetics, where mutations in specific genes can lead to the onset of the disease. And these are the ones we are focusing in this part of the project. It is important to mention that there is currently no treatment that cures or stops the progression of the disease. In this scenario, what we try to do in the lab is to develop tools for testing potential drug candidates to treat the disease. For this, we will need to have neurons from the patients, and this is hard to obtain. So what we do instead is we take a skin sample from the patients and we isolate a type of cell called fibroblast. In here, what you can see is a representative image of how these cells look in a bright field microscope. Then, to these cells, we perform a technique developed by the Yamanaka's lab, which consists in reprogramming these cells into a pluripotent cell, a cell that has the capacity of forming different cell types. In other words, what we do is we transform these cells into the ones that are present during the early stages of development. In this picture, what you can see is how these cells, in this case forming a colony, look under the microscope. From this point on, what we can do is we can guide them to become the cell type we like in a process called differentiation. And we do so first into a neural stem cell state. And this is the precursor of the final neurons that we want to obtain. In this slide, what you can see is how the process in some short images looks like. So we perform a purification of the cells by passaging, and after we achieve a certain passage, we control that these cells are expressing a specific set of markers. Afterwards, we push them a bit further and we obtain fully differentiated neurons. In this image, what you can see is how different markers, such as a nuclear marker, an overall neuronal marker, and the marker for dopaminergic neurons that we are using, DH, looks like. Since we are focusing on the development of Parkinson's due to genetic mutations, we also need to account for the effects of these mutations. And for this, we generate what is called an isogenic line. That is a copy of the cell where only one relevant mutation is modified. There are different ways of doing so, but in this project we develop a fast way of controlling the modification of both genetic copies of a specific gene at the same time, because most of the genes that we have two copies. And we do so by providing a DNA construct that not only carries a genetic mutation that we want to introduce, but also a fluorescent reporter that allow us to detect which were the cells that were correctly modified. By using different types of fluorescence, we can select those ones that presents both events of modification. In this case, we're using a green fluorescent and a red fluorescent, so whenever we see that the cells are shining yellow under the microscope, this means that this cell has both copies modified at the same time. In this case, what we also include is another fluorescent, in this case a blue fluorescence, that allows us to detect when a cell was incorrectly modified. So normally in this process, What we do is we first select the cells that were correctly modifying by flow cytometry, just by selecting the ones that are not expressing the blue fluorescence, and then afterwards we purify the cells that are having both fluorescence expressed at the same time, so the red and the green making a yellow cell. Once we have a stable culture with a purified cell population, what we do is we just remove the fluorescence, leaving in the genome of that specific cells the mutation that we wanted to introduce. If you would like to see more information, please refer to this publication. In order to study Parkinson's disease, we don't only need the cells. We also need that these cells form a 3D architecture that resembles a human tissue. For this, we use the OrganoPlate from the company Mimetas, that characterizes on having an array of chips that allow 3D culture, maintain flow of media, and can contain multiple cell types, and is fully compatible with automation systems and high-content screening microscopes. These chips consist of four chambers: the shell inlet, where we deposit the cells in a matrix, a perfusion inlet, where the media flows to the perfusion outlet, and an observation window. If we would make a cross-section of the observation window, what we can see on the right is the matrix containing the cells growing in 3D, and the channel where the media is flowing. For this particular set of experiments that I'll be explaining, we use 8 different cell lines. This accounts for the cells derived from healthy individuals and from Parkinson patients that are carrying a specific mutation in the LRRK2 gene. In this case, the mutation that we are analyzing is G2019S. As isogenic controls, what we did is we introduced a mutation in the healthy individuals, and we corrected the mutation in the patient-derived lines. The neural stem cells were seeded in the OrganoPlate and cultured up to 6 weeks. Afterwards, we performed immunofluorescence stainings to quantify the proportion of and the type of neurons that we are observing, and we did so in an automated manner with image analysis algorithms that we developed in-house. If you would like to see more information about experiments, please refer to this publication. One of the first results we observed was that the amount of dopaminergic neurons in the patient was reduced. Not only the amount, but also the morphology of the neurons was altered after 6 weeks of culture. What you can see in this panel is a comparison between what we see in the healthy individual and in those cells derived from patients carrying the G2019S mutation. And we can see a marker known as TH that is representing the type of neuron that we want to analyze, the one that is lost during Parkinson's disease. And on the bottom of the graph, what you can see is how these neurons are branching. So it's the level of complexity and three-dimensional arrangement that these neurons have. And we also see that this branching is reduced in the case of the patients. We also checked the status of the mitochondria in these cells. And what we observed was that the quantity and the morphology was altered in the neurons derived from Parkinson patient cells. In this panel, the staining that we perform at 3 weeks of differentiation, what you can see is the comparison again between the healthy line and the lines derived from Parkinson patients. And you can see that the amount of mitochondria as well as their morphology is altered in the case of the patient. As a proof of concept for a drug screening program, what we did is we used a known compound called inhibitor 2, and after testing it, what we saw was that the levels of dopaminergic neurons were restored in the case of the patient. When we compared the effect of the treatment against the effect of the gene correction, we saw that the effect of the inhibitor 2 was stronger than the modification in the genome that we did. And looking more into detail into this phenomenon, what we saw was that the introduction of the mutation in the healthy line was enough for triggering the problems of differentiation that we observe in the patient. But whenever we corrected the gene mutation in the patients, this correction was not enough to restore the phenotypes that we observe. This points to the fact that maybe there is some other mutations in the rest of the genome in the case of the patients that are causing this phenomenon. This is what we refer to as the background, the genetic background of the patient. In conclusion, in this part of the project, we optimize a culture of patient-derived neurons carrying the LARC2 C289S mutation in 3D microfluidics. We implemented an automated image analysis algorithm which revealed robust phenotypes, and we also restored the reduced dopaminergic differentiation as a proof of concept with the treatment with inhibitor 2. And this restoration of the reduced dopaminergic neuron was something that we observed only after the treatment with the drug, but not with the gene correction. With this, I would like to thank all the members of the 4 institutions involved in this part of the project, the past and current members of the Developmental and Cellular Biology group, as well as the entire SysMedPD consortium, and to you for taking the time to listen to this presentation.